The present study concerns the validation of ResEP, a device for elimination of antibiotics from liquid samples undergoing microbiological analysis in tissue banking. The study, performed by AlkiLife, was presented at the 27th EUSOL-C meeting in 2015 in Venice. During our studies, we realized that the use of antibiotic cocktails in tissue decontamination can cause the presence of antibiotic residues both on tissues and in processing liquids. These residues could induce bacteriostasis of the microorganisms possibly present in the samples to test, thus causing false negative results during microbiological analysis. In addition, European and US pharmacopoeias clearly indicate that the sterility tests must be performed after a complete removal of any possible antimicrobial agents. For these reasons, we have performed several studies concerning this topic over the past 6 years. Among them, there is a study performed in collaboration with 2 Italian cardiovascular tissue banks, whose results have been recently published. With all these studies, we aimed at obtaining a device capable of removing antibiotic residues from liquid samples undergoing microbiological analysis. In particular, the device had to be innocuous for the bacteria eventually present in the sample, thus allowing their detection. Finally, the device had to be compatible with the currently adopted microbiological analysis methods in order to be easily added to the tissue bank routine. Therefore, we developed the ResEP medical device that you can see represented in the picture on the top of the slide. The device is composed by a syringe containing a resin mixture able to absorb and then remove antibiotic residues from the liquid samples to test. The needle is provided with a safety cap that guarantees safe disposal after use. The device must be used under aseptic conditions as follows. ResEP syringe and needles are removed from the sterile pouches. The device is uncapped and then the needle is screwed. The sample to test between 3 to 9 ml is drawn with ResEP, and then the device is mechanically shaken at room temperature for 20 minutes. Finally, the sample is ejected in the vial with growth broth for microbiological analysis. The aim of the present study was the validation of the ResEP medical device. The process consisted of 4 steps. First, we checked whether the device was effective in removing antibiotics from liquid samples. We verified the compatibility of the components of the device with the bacteria eventually present in the sample to assure that the medical device did not interfere with their survival and growth. Further, we performed the method suitability test in compliance with the European Pharmacopoeia to verify that the sample to be tested, that in this case is an antibiotic cocktail, did not interfere with the results of the test. Finally, we asked the tissue banks to verify whether the device was compatible and could be used together with their standard operating procedures. The first validation step consisted in the evaluation of the antibiotic removal from 4 antibiotic cocktails, 3 cornet storage media and 1 tissue decontamination medium, namely EUSOL-C, Tissue C, Carry C and Base 128. The samples were treated with ResEP at room temperature for 20 minutes and then analyzed by HPLC. The test was performed on 3 and 6 ml of each antibiotic cocktail before and after tissue preservation. These conditions were selected considering that the presence of tissue and hemathic components could reduce the capability of the medical device to remove antibiotics due to the presence of more components in the sample. To evaluate the possibility that the components of the device interfere with bacterial growth, inoculants of 1 to 10 CFU and 10 to 100 CFU were treated with ResEP at room temperature for 20 minutes and then seated in agar plates. CFU growth was assessed and quantified after 24 hours. To test the suitability of the method according to the European Pharmacopoeia, we verify that the same inocular 1 to 10 and 10 to 100 CFU contained in antibiotic cocktails, EUSOL-C, tissue-C, carry-C, and base 128, were able to generate turbidity in growth media within 5 days after ResEP treatment. Finally, we asked the tissue bank operators to evaluate whether ResEP was suitable and compatible with their standard procedures in terms of tested volumes, processing time, and ease of use. In particular, 4 systems were compared: BACTEC, BACT-ALERT, HBNL, and the direct inoculation method. The figure shows the results of the antibiotic removal from 3 and 6 ml of the first analyzed cocktail. The analysis referred to the antibiotic removal from the pure cocktail and from the cocktail after corneal storage. The initial antibiotic concentration was similar in pure cocktail media and in cocktail after corneal storage. The ResEP treatment completely removed the antibiotic in all the tested conditions. The same results were obtained for cocktail 2. The whole initial antibiotic and the antimycotic content was completely removed from 3 and 6 ml samples of pure cocktail and cocktail after corneal storage. The same was observed for cocktail 3. All antibiotics and the antimycotic initially present in the media were completely removed after the ResEP treatment. Similarly, all antibiotics and the antimycotic present in cocktail 4 were effectively removed both before and after cardiac valve decontamination. The device resulted perfectly innocuous for the tested inocula, since a complete recovery of even 1 to 10 CFU was observed for all the European Pharmacopoeia reference strains. Despite the presence of an antibiotic cocktail in the bacteria inocula, after ResEP treatment, turbidity of the media, indicative of microbial growth, was observed within the 5th day as required by the method suitability test, according to the European Pharmacopoeia. Finally, ResEP was given to tissue bank operators to test whether it was suitable to be used together with BACTEC, BACT-ALERT, HBNL systems and culture media of the direct inoculation method. The results indicated that the volumes of samples resulted compatible with all the tested systems, the processing time was compatible with all the tissue bank procedures, and the device was simple and safe to use. In conclusion, we consider ResEP validated based on the results of our study indicating that ResEP is able to remove easily and quickly antibiotics from all the tested liquid sample, base 128, EUSOL-C, tissue-C and carry-C, showing high specificity for the antibiotics contained in these cocktails. ResEP is innocuous and perfectly compatible with bacterial survival and growth and allows a complete microbial recovery of even 1 to 10 CFU for all the tested strains. The direct inoculation method with ResEP resulted to be suitable according to the European Pharmacopoeia requirements. And ResEP can be used together with the currently adapted microbiological analysis methods to eliminate antibiotics from the samples before the test.